So good afternoon, everyone. It's a pleasure to have you all present here with us today. I'm very pleased to introduce to you Philippe Boileau, in case you've not already come across him. He's located here at the core at least a part of the week. Philippe recently joined the Department of Epidemiology, Biostatistics, and Occupational Health and Medicine. He's jointly appointed in both these departments. And so everybody wants to have a piece of Phil. This is what's been happening the last few months. Philippe, as it happens, is a Montreal native. He studied here in Concordia University to start with. This is where he did his undergraduate. And then he went away to the University of California in Berkeley and thankfully found his way back. So today, Philip is going to talk to us about something that I think, often ignore in statistics. We're often so focused on the mean that we forget about the variance. And, if any of you have done meta-analysis, you know that our focus is on the pooled mean statistic. This is what we're after. And we forget about between study heterogeneity. And I think that theme is in many settings. So you're going to help us to turn our attention towards the variance for a change. And this term assumption lean is entirely new to me. So I hope you're going to shed some light on where it's coming from. I've heard of non-parametric, I've heard of distribution free, but this is new. And apparently, Philippe is going to tell us how it brings together causal inference, machine learning, and semi-parametric methods. So yeah, I'm very excited to hear about this. Thank you.
Thank you very much for the introduction. So I'll briefly touch on what is assumption in statistics, but if that definition is not enough, we could always go over it during the question period. So this work was done with a team of statisticians and clinician scientists. The statisticians include Hanizaki and Mireille Schnitzer from the University of Montreal, and then the clinician scientists include Gabrielle Lillekite, Nicholas Nielsen from the University of Lund, and our very own Patrick Lawler at the RI. So I'll talk a little bit about my motivation. So most of my methodological work that I do right now is actually motivated by some challenging problems that I encountered while working as a data scientist and consultant in the pharma industry. And the one that I'm most interested in revolves around this major issue that we encounter in the drug development pipeline, which I'm going to grossly oversimplify here. And so basically the drug development pipeline from a high level can be split into three different steps. The first is basic and preclinical research, where you find a drug that might be used to treat a specific indication. You then have in-human trials, and if these are successful, you move on to regulatory approval and launch of that drug. And I'm going to focus in particular on the clinical trials portion of this drug development pipeline, because that's where most of the interesting data analysis takes place. And as a statistician, that's what I want to focus on. So for those of you who are not familiar about this clinical trial or drug development pipeline and how it relates to clinical trials, usually what happens is in your basic preclinical research, you identify that drug, as I mentioned, and then you have your first in-person trial, where you're trying to not identify whether or not the drug actually works, but you're just trying to find a good, reasonable dose that is tolerable for humans to take. Assuming you can find a dose that works, you then move on to a slightly larger phase 2 trial. This can be randomized, but it isn't always. And here the goal is to really determine the safety and tolerability of this drug and maybe get some preliminary efficacy signal. And if that's successful, what you could then do is run a phase 3 trial. These are large randomized studies where the primary goal is to compare the efficacy of this new drug against the standard of care. All right, and if this is successful, then you can move on to actually selling your drug on the market, assuming that it's approved by regulators. I'm having some slide issues, but there should be a box over here that shows that next step. Now, the trials themselves increase in the required resources from stage to stage, not only because investigators and sponsors are putting in their resources, but also because patients, the size of the samples increases from trial to trial. And so the worst thing that could really happen, or one of the worst things that could happen in this pipeline, is that you run through this entire process of the phase 1 and phase 2 trial. Things go well, you run your phase 3 trial, and at the very end, at the readout, you find out that your drug is actually not as efficacious as you thought. And so what's happening over here basically is that you have wasted resources throughout this entire pipeline that could have been better distributed elsewhere. More importantly, I'd argue there are patients involved in this pipeline who could have actually received other investigatory therapies that might have actually benefited them for real, based off of the efficacy measure in this phase 3 trial. And so you might think this doesn't happen very often, given that this is a rigorous pipeline, at least scientifically. But from some quick research I did, I found that it's actually really not the case. In fact, almost half of all trials identified in public databases and commercial databases between 1998 and 2000 were reported as null, having null findings. And so there really is a need to understand why this is happening and try and mitigate the risk of these failures in these late stages. Again, to benefit patients and also just to distribute resources in a more effective manner. And so there are many reasons why this might happen. The one that I'm most interested in is this idea of there being heterogeneous subgroups that drive these results. All right. So over here, I have the sketch of a phase 2 randomized trial where we have 2 treatment arms, treatment arm itself with novel therapy and the control arm with the standard of care. Now, these dots represent patients and they make up different clusters based off of 2 different baseline covariates, which are either unmeasured at the time of collection or just not accounted for in the design of the study. All right. Now, it's not unreasonable to think that these clusters also form patient phenotypes. And these patient phenotypes respond differently to the drugs that are being administered to them, right? So in the top left corner over here, we have a subgroup that benefits more from this novel treatment than the standard of care. The same is true for this subgroup in the bottom left corner. In the bottom right corner, we have a small subgroup that actually has a negative reaction to our novel therapy, but responds very favorably to the standard of care. Now, we're not going to get to see these outcomes per subgroup. We're only going to see the marginal effect. So if we take the weighted average across these subgroups, our final story or summary of this trial is that this new medication is slightly better than care. So we're happy with this, and we'll move on to phase 3. All right. And for simplicity, we'll assume that we're looking at a similar endpoint. And that means that we have basically identical or very similar patient phenotypes appearing in this trial, which is slightly larger, like I said before, but now with different proportions. Maybe because we're recruiting from different centers, or there's a shift in the patient population, whatever it is, proportions have changed. Each phenotype has the exact same rule or kind of outcome, in that some phenotypes favor the new treatment, some favor the old treatment. But again, we're not taking into account these baseline covariates. And so when you take the weighted average, you might end up with something like this, where now the standard of care is outperforming the new medication. And so this would result in a null trial. This would be a negative outcome for patients as well as for sponsors and investigators. So what's actually happening over here? Well, it turns out that this baseline covariate 1 on this x-axis over here, now plotted over here, is a treatment effect modifier. And the reason this is because if you plot out the predicted outcomes on average across these patients, depending on their baseline covariates, you see that patients who have larger values of the baseline covariate have better outcomes if they were assigned to the standard of care, the control arm, versus the treatment arm. If patients have lower values of the baseline covariate, then the opposite is true. And so you could really use this information to design a trial, right? Had access to this kind of plot or these results, or you just knew that baseline covariate 1 was a treatment effect modifier, then maybe you would restrict your trial participants only to this group of patients over here. And in your guidelines, you would note that patients who have large values of baseline covariate 1 should receive the standard of care. All right. Now, the challenge is that it's not always easy to figure out what these effect modifiers are. Standard procedures that do this exist, and they can do so quite well, but they rely on actually having access to the effect modifiers in the first place. They kind of have to be known a priori, but at the very least collected in your data. But in practice, we don't always know what these effect modifiers are. They're not always collected due to potentially resource constraints. And then 3, even if we do know them, it's very possible that they're mismeasured, simply because they're hard to measure or there's measurement error in the process of actually collecting the data. So the question we're going to talk about today is whether or not we can still uncover evidence of heterogeneity, even if we don't have access to these effect modifiers. And so I'll stop now to mention that I'm really painting this as being important in the case of late-stage clinical trials, but effect modification appears in really all different kinds of settings, right? So you could think of even having a successful phase 3 trial, but now wanting to apply your findings to a wider patient population that's also heterogeneous. Well, you're going to want to make sure that you have some understanding of your effect modifiers to see how the actual population will react on average to your medication. You could also think of trying to deliver optimally new public health interventions to heterogeneous communities, or tailoring delivery care models to your local hospital environment, or even optimizing policy and health economics, for example, to make sure that your interventions work as intended in various communities. All right, so in all of these settings, it's important to have a sense of what the effect modifiers are. And again, in all of these settings, you don't always have access to these effect modifiers or even know what they are everywhere. All right, so now I'm going to formulate a little bit more rigorously, and we'll start with considering an idealized data set. All right, so over here, what I have is a spreadsheet that you might open up if you can run a perfect prior log group experiment, where either you're in a randomized setting or you have observational data. Of that study design, you're always going to have access to P baseline covariates, which I'm representing by W. And these baseline covariates can be of any type. They could be binary, they can be continuous, they can be category dummy variables. That's totally okay. We have a binary treatment indicator A. 1 is for the treatment arm, 0 is for the control arm, or 1 is for the exposure, 0 is for the unexposed. And then finally, we have access to potential outcomes. And this is where I'm talking about, or where the idealized component of this idealized data set comes in. All right. So the potential outcome in this column over here, Y0, represents the outcome that every individual would have had they actually been assigned to the control condition, all right, or the control arm. And then we have the potential outcome on the far right over here under treatment, which is the outcome that these individuals or observations would have if they were actually assigned to the treatment group. All right. Now, we never actually have access to these two columns in reality. We'll only get access to the outcome corresponding to the treatment assignment. So, for example, observation 1 is actually assigned to the treatment arm, and so we're not going to have access to Y0, we'll only have access to Y1. Similarly, for observation 2, they're assigned to the control arm, and so we only have access to this red square over here, which represents their outcome under control. All right. Nevertheless, if we continue with this idealized data set for a moment, we'll see why it's useful in the context of detecting heterogeneous treatment effects. All right. But we'll start with just measuring treatment effects more generally. All right. So if you have both of your potential outcomes, what you can do is compute the individual treatment effect for all individuals. That is, you take the outcome under treatment minus the outcome under control. And you get to have, in this far right corner, that individual's treatment effect. And you could determine for each individual whether they should be assigned a treatment or optimize their outcomes. All right. Now, if you have a very large data set and you have this idealized data set, you're not going to only want to rely on this entire column of data. It's hard to summarize. Instead, you want to come up with a simple story to report whether or not this treatment is working. And to do that, we rely on this causal estimand or causal parameter called the average treatment effect. And that is simply the average of this column if you had access to population data. All right. Now, as I was saying, this ATE is a summary of the patient treatment effect. It just gives you the sense of whether or not, on average, patients benefit more or less from being assigned to the treatment or control group. But if you want to detect heterogeneity, we have to understand how this column changes as a function of your baseline covariates W. And so arguably the most popular parameter for doing this is something called the conditional average treatment effect. All right. It's really just your average treatment effect, so the average of the difference of your potential outcomes as a function of W. Or in even simpler terms, it's like regressing your outcome, your individual treatment effects on all of your baseline covariates. All right. So if you actually had access to this data, you could literally just fit a linear model where this difference of potential outcomes is your outcome and use your W's from your baseline covariates to fit your model. All right. As I mentioned before, even though this is a very popular parameter, it has limitations, all right? And that's that it's conditional on W. That means you actually have to have access to true effect modifiers in your set of baseline covariates to detect effect heterogeneity. And you might think, well, instead of working carefully to try and identify these effect modifiers, maybe I just take all the data I have on my observations, take the entire kitchen sink and throw it at my problem, all right? In which case you'd end up with a high dimensional regression problem where W is potentially much larger than your total size of your data set. And here we run into issues again, in that this kind of estimation procedure is just very hard. And so trying to take this approach will actually rely in unreliable inference in general, and very strong simplifying assumptions about how your data is being generated, all right? So we're going to try and address these limitations by defining another target of inference, another causal estimate, another way to summarize the relationship between these two columns over here that allows us to detect heterogeneity. All right. So I started off this example by taking a heterogeneous treatment effect as assumed, as being like the way we would always start off with these problems. But let's take a step back and think about what a homogeneous treatment effect looks like, all right? And that is where every individual has the exact same individual treatment effect over here, meaning your outcome under the treatment arm is nothing more than your outcome under control plus some constant shift for all individuals, this delta over here, all right? Now in this setting, this delta is actually equal to the ATE because the ATE, remember, is just equal to the population average of this column. And so in this particular example, the ATE is equal to 2. So from this idea, can we actually investigate whether treatment effect homogeneity is reasonable instead of immediately jumping to the conclusion that a treatment effect is heterogeneous and we should find the root cause? And the answer is we can do this, all right? And it's based off of this simple relationship that we found while thinking about this problem. And that's that if you truly have a homogeneous effect, that is your outcomes under your potential outcomes under treatment are just a shift of your potential outcomes under control, then it turns out that the variance of your two potential outcomes are exactly identical, all right? But if the variances of your potential outcomes are different, meaning their difference is equal to non-zero or the ratio is not equal to 1, then it must be that you don't have a homogeneous treatment effect, all right? And so the bottom line here is that we can actually uncover evidence of effect heterogeneity through a simple contrast of our potential outcome variances. By contrast, I mean differences or ratios, all right? And we could do all this without having to account for any W's, right? There's no W's here. Now, there's a nice proof of this. I'm not going to show you the math. Yeah.
So, probably an ignorant question, but I just want to make sure I'm following what you're saying. This looks very elegant, and you are taking out the covariates, which you say are problematic and can make the problem multidimensional and so on. But here, isn't it true? We don't know what y1 and y0 are.
That is a great point to raise, and I'm going to address that in the future slides. But yes, so even though we've dealt with the W issue, we're still in this fantasy land where we have access to the outcomes under both treatment and control. I'll ask you to bear with me just a little bit longer, and we'll see how we could actually resolve this issue. All right, while still not having to rely on the W's. All right. Now, like I was saying, there's some nice math proof here to show that this is true. I'll show you a visual to provide you with intuition instead of showing you the math proof. Okay. So let's start off with that first line where we assume that we have some treatment effect. All right. And so in this plot over here, we have the distribution of individual treatment effects under that setting. Now, it's not much of a distribution because everybody has the same exact ITE at this specific line over here, all right? And so what we can do is actually plot the individual's outcomes under Y0 and under Y1, all right? Again, under this homogeneous treatment effect assumption. And so what this really means is that I could take, for example, an individual who has a outcome under control of about equal to 9, and then I would know that under treatment, they would have had a value or an outcome equal to 11, simply because everybody has the exact same treatment effect. And this is true for any single individual that I'd pick out from this distribution. If you have a value of 10 under control, your outcome is equal to 12 under treatment. If you have a value of 12 in your outcome under the control, then you have a value of 14 under treatment. All right. Now, what's nice about plotting these distributions is you see that there's really just a shift going on. And so in fact, they actually have identical variances, identical spreads in these two densities. All right. So what about a setting where you have these two distributions, but they don't have equal variances? All right. Is this indicative of a heterogeneous treatment effect? Well, in this setting, you can't really eyeball an easy rule to see how patients shift from their outcome under the control versus their outcome under the treatment. Maybe individuals with a value of about 10 under the outcome have a value of about, I don't know, what's that, 18 under treatment? Or maybe it's a slightly smaller value of about 6 or about 15. All right, there's really no easy way of knowing purely based off of this plot. It turns out that if you actually have access to the individual's data and you compute the ITE, then you get something with an actual distribution. So unlike this line over here, there is a spread around the individual treatment effect. And this symbolizes that there's heterogeneity. And so basically the bottom line is that if you see these two distributions, they have different variances, then there's a guarantee that you have this kind of spread over here, and therefore you have to have a heterogeneous treatment effect. Right? Okay. Now we want to capture this in some way. We want to summarize this. We can actually plot our distributions. And so we're going to think about two different, or a set, I should say, of different causal estimates, causal parameters that summarize this relationship. Now, the first that we've considered is the absolute differential variance, which is simply the difference of the standard deviations of these potential outcomes. All right. We could have done the difference of the variances, but what's nice about this parameter is that now the units are on the same scale as the actual outcomes themselves. And so it's a little bit easier to, right? Talking about squared units is always tough. On the other hand, if you're interested in some relative metric of differential variance, then we take the ratio of the variances. And so this gives you a sense of how much the variance under treatment is inflated or deflated compared to the variance of the outcome under control. The bottom line is that with these parameters, we can define formal hypothesis tests, all right? Where, for example, in the case of psi c, we have under the null hypothesis of a homogeneous treatment effect that psi c has to equal to 0. Because again, the variances, or equivalent to standard deviations, are equal to one another. And so this is equal to 0. But as soon as it's non-zero, then you have to reject that assumption of homogeneity of effect. The same is true for this lambda over here. All right? Now, as Nandini pointed out, it's all nice and dandy to have defined these parameters and have this hypothesis test, but we can't actually define or estimate these guys in practice, right? Because we don't actually have access to these shaded out columns in our data set. We only have access to the outcome that we get to observe. So again, your outcome under, for this individual over here, they're assigned to the treatment group. So the outcome we observe is actually their potential outcome under treatment. Observation number 2, they're assigned to the control group. So the outcome we observe is going to be equal to their outcome under the potential outcome under control, and so on and so forth. Oh, can we still identify or estimate, that is, these causal parameters that depend on potential outcomes with our observed data? And the answer is yes. It turns out that under very standard conditions in the causal inference literature, we could actually write out our causal parameters in terms of observed data parameters. The math is, there's a lot going on here. Don't worry about it. The bottom line is that so long as we have conditional exchangeability, so there's no unmeasured confounders, that we have consistency, meaning that the observed outcomes correspond to the correct potential outcomes, and finally positivity, that all of our observations can receive either treatment, then we are able to estimate these causal parameters. Now, in general, these conditions are not verifiable. We have to make some pretty strong assumptions about how data is generated based off of substantive area expertise in observational settings. If you're in a randomized controlled trial, it turns out that these conditions are satisfied by construction, by design. And so in an RCT, you can estimate those causal parameters even though you don't actually have access to your potential outcomes. All right? So we made the connection now from this idealized data set to real life, and it turns out we can still estimate those parameters under conditions that are at least satisfied or guaranteed to be satisfied in an RCT. So now the question is, how do we actually estimate them? all right? Now, there's plenty of ways to do this. We've relied on causal machine learning estimators, however, all right? And so we proposed 4 causal machine learning estimators per differential variance estimate, so the absolute differential variance and the relative differential variance. So we have TMLEs, one-step estimators, and then cross-fit versions of these estimators. Now, these estimators are labeled now more and more in literature as assumption lean because they don't actually rely on any assumptions about how the data is being generated. Like, for example, linearity. So these are going to be correct, so to speak, even in non-parametric models or in complex models where you don't want to make any simplifying assumptions. It's also important to note that because they're non-parametric, you could actually flexibly adjust for confounders, if any. It's not the case in an RCT, but in an observational study, this is important. And finally, if you are in an RCT, you can still adjust for prognostic variables, which again is important from a precision point of view, and based off of FDA guidance on how to actually estimate estimates in clinical trials. So these causal machine learning estimators actually internally rely on estimating 3 nuisance parameters under the hood, so to speak. The first is a propensity score, so the probability of receiving treatment as a function of baseline covariates. The second is an outcome regression, so the average outcome as a function of baseline covariates and treatment assignment. And then finally, the squared outcome regression, which is the average squared outcome as a function of baseline covariates and treatment assignment. And again, because these are causal machine learning estimators, the machine learning part comes into play because we could estimate these nuisance parameters or these components of our estimators by using, if we wanted to, flexible machine learning estimators. So we're talking about something as simple as lasso regressions all the way up to neural networks or forests, you name it, we can use it and we can still get valid inference. Now, the reason people like causal machine learning estimators is that they tend to have 2 very nice properties, all right? The first is double robustness. And the idea here, in the case of RCTs in particular, is that if you know the propensity score, which you do, it's the way that you randomize patients to your treatment arms, then these estimators are guaranteed to be unbiased in large samples, right? Remember, we have to model outcome regressions. What this says is that even if we misspecify our outcome regressions, we can still get unbiased estimates. All right, so that's a nice property in large samples. But the real kicker in this is asymptotic linearity. So again, I have a bunch of jargon over here, but the bottom line is that in clinical trials, because the propensity score is known, these estimators are asymptotically normally distributed, all right? And so this gives us a way to actually perform valid hypothesis tests, even when the outcome and squared outcome regressions are some complicated relationship, all right, that might not be easy to estimate in practice. And so again, just to drive this point home, this nice asymptotic result, this statistical theory tells us that we can perform formal hypothesis tests for heterogeneous treatment effects in RCTs without having to make strong assumptions. All right. And so again, we can go back to our idealized hypotheses that we were talking, hypothesis tests we talked about. And it turns out we could run them in observed data with nice traditional statistical properties, like, for example, type 1 error rate control. All right. Okay. So then we want to make sure that these asymptotic results kick in practice. We ran a few simulation studies, namely one for the assessment of double robustness, one for asymptotic linearity. I won't show them to you here. I'll just let you know that it works, all right, for most reasonable settings. What I think is interesting to show is this third simulation study. We wanted to assess the impact of measurement error on our ability to detect heterogeneous treatment effects and compare it against some of the popular CATE-based estimation procedures that are currently used for identifying heterogeneous treatment effects. All right. So the setup is we have a simulated randomized control trial. And to keep it simple, we only have one treatment effect modifier. All right. Now, this treatment effect modifier is mismeasured to some degree that we specify in different scenarios, ranging from wholly misspecified, meaning it may as well not be measured, to having no mismeasurement whatsoever. We then evaluated our machine learning estimators against competing estimators like the causal forest, all right, which is a very popular approach in the literature, in terms of the empirical power to detect heterogeneity. And so these are the results, all right? So there's a lot going on, but basically each panel represents a different scenario with different amounts of measurement error. This is zero measurement error. About a third of our effect modifiers are mismeasured. 2/3 of observations effect modifiers are mismeasured. And finally, all all of the effect modifier measurements are mismeasured, which is essentially equivalent to not having measured the effect modifier at all. On the y-axis, we have empirical power. On the x-axis, we have sample size. And finally, these different lines and dots represent our different estimators. So we have, in the different colors, the estimators that we've derived for the absolute differential variance estimand. And in black, we have the causal forest. All right. And so stepping through this one by one, in the setting where there is no error at all in our measurement of our effect modifier, we see that our causal forest estimator does a really good job of identifying or basically telling us, making the decision that there is in fact effect heterogeneity, all right? In fact, much better than our proposed estimators in smaller sample sizes. Whether this, for most estimators at least, kind of gets to the identical solution most of the time at like sample size of 500. But as you increase the error in our measurement of our effect modifier, you see that the performance of the causal forest over here decreases. And it decreases more and more as measurement error increases. And so in fact, in this setting over here, where there's 33% mismeasurement or measurement error rate, 3 of our 4 estimators actually outperform the causal forest at a sample size of about 100. But then the causal forest quickly takes over at a sample size of 200 and then 500. Here, we see that the causal forest struggles much more until about a sample size of 1,000. All right. And then finally, when there is absolutely no signal from the actual effect modifier, the causal forest is just spitting out false. Well, they're true positives, but in its head, they're actually false negatives. It's just, false positive. It's just controlling a type 1 error rate. Yeah. Oh, sorry. So OS is the one-step estimator. TMLE is a TMLE, and then the CF-OS and CF-TMLE are cross-fitted versions of these 2 different estimators. So there's some differences, but it's not really worth diving into the weeds just yet. They all tend to work relatively well. Well, I'll note that the CF-TMLE tends to perform slightly worse in a specific setting. I'll also note that the underlying relationship between the outcome and the covariates in this clinical trial was very nonlinear, well, simulated clinical trial. But we chose to estimate our outcome regressions using simple linear regression models, all right, to mimic the guidelines for ICH, which say that you should use simple linear or just linear regression models. Really simple. Don't rely on machine learning estimators for these nuisance parameters, just to keep things simple and not to inflate variance. So we actually penalized ourselves a little bit. We ran this exact same experiment using flexible machine learning estimators for nuisance parameters. And in fact, in that situation, all of our estimators, so these colored guys over here, have empirical power above 80% across the board in every single panel. Now, the last thing I want to highlight is that basically there's no difference between any of our estimators' performance regardless of the panel, showing that we don't actually rely that heavily on having accurate measurements of our effect modifier to detect effect heterogeneity. All right, and now on to the application. So for those of you who are not aware, targeted temperature management is an intervention that induces a mild controlled hypothermia between 36 and 72 hours, usually following cardiac arrest, all right, where a patient goes into a coma. Now, the idea is that this cooling will actually reduce neurological damage following ischemia or reduced blood flow to the brain, because a lot of the damage is actually felt following the restarting of circulation of the blood back to normal levels. And so somehow cooling reduces or is at least thought to reduce the amount of damage neurological to these patients. Now, unfortunately, like when this was first put into guidelines for patients who experience an out-of-hospital cardiac arrest, it wasn't clear what temperature was the best temperature to cool patients to. And so Nicholas, the co-authors on this paper, was a PI for 2 trials. The TTM trial, which was the first of these 2 trials, followed by TTM2. So TTM1, patients were randomized to either 33 degrees Celsius or 36 degrees Celsius cooling. And in TTM2, it was hypothermia at 33 degrees Celsius versus normothermia, which is just maintaining a body temperature below 37.5 degrees Celsius. And Nicholas was interested in determining whether or not there was any difference in these patients' survival up to 6 months. All right. Now, either trial found a significant difference in the outcomes of these patients, which was the primary outcome, depending on the choice of target temperature. All right. And so the guidelines were modified to reflect this. And so now the general guidelines just say keep patients' temperature below fever, so below 37.5 degrees Celsius. But in August of this year, there were some critical care physicians who hypothesized that, in fact, there might be a subgroup of patients who benefit more from lower temperature, targeted temperature management, especially if they had signs of severe neurological injury or prolonged ischemia duration. All right. And so there's some in-progress work in Patrick Lawler's group that are attempting to detect whether or not these effect modifiers or effect modifiers related to these kinds of patients, these patient phenotypes, truly modify the effect of targeted temperature management. But so far, at least in a reduced set of variables that we're looking at, we haven't found anything. It's been null results. All right. And so this could just be that there are no effect modifiers, but it might also be due to the fact that some of the effect modifiers are approximated in the clinical trial data. So for example, time to advanced life support is a potentially important effect modifier, but it's actually approximated based off of hospital notes, right, in this trial. Another alternative is that we're simply missing some actual effect modifiers from this data, and so we would never even be able to detect heterogeneity using traditional methods. All right, and so we decided to run an absolute differential variance analysis as a sensitivity analysis for this, where we treated the primary endpoint as days of survival from intervention as a continuous outcome because there's virtually no right censoring, so we wanted to improve power. And then we inferred our causal estimate using our one-step estimator and our TMLE, so the non-cross-fitted versions, because the cross-fitted versions perform slightly less well, as we saw in the simulation studies. All right. And then because we had access to a number of baseline covariates, we decided to adjust for these baseline covariates to increase precision of our models. And again, we used linear models, all right, for our nuisance parameter estimators, with the exception of the propensity scores of Ferdinand. So we plugged in the propensity scores to guarantee some nice statistical properties for our estimators. And so these are the results. Up top, we have the results for our TTM trial using both the one-step and TMLE. And so our estimated difference in standard deviations of the potential outcomes is 3.9 days and 3.6 days respectively, with relatively small standard deviations, but still p-values that are non-significant at the 5% level. All right. In the TTM2 trial, where we had almost twice as many participants, the intervention was slightly different, but now we have estimates that are much closer to 0, equal to 0.9 days. But because the standard errors were so small, we actually end up with p-values that are below the significance threshold that we set. And so at the end of the day, what we've done is we've rejected the null hypothesis. We failed to reject the null hypothesis of a homogeneous treatment effect in the TTM trial, but we reject that null hypothesis in the TTM2 trial. All right. Now that's all fine and dandy, except that this absolute differential variance estimate of 0.9 days just means that there's a 0.9 day difference in the standard deviations of outcomes across both treatment arms. And remember, the outcome of interest is 6 months survival or 180 days. So really this 0.9 estimate probably isn't really that clinically meaningful, even though we have a statistically significant result. So the bottom line here is that although we have evidence of a heterogeneous treatment effect, it's likely not clinically meaningful or actionable. All right. So this is the first real application of this methodology in this data set. Oh, yeah.
So what was the difference in sample size between these two, TTM and TTM2?
Great question. Yeah, so TTM, the first TTM was about 900, and TTM2 was 1850, I think. So almost twice as large, which might explain the more precise estimates or the smaller standard errors, but also it's important to keep in mind that we're actually comparing different interventions. But still, it gets at the question that these clinical care physicians posed in August as to whether or not there is evidence of effect heterogeneity. And our answer here is there is, but it's very small and likely not practical to address. All right. And so just to recap what we've done, we've developed some new causal parameters for detecting heterogeneous treatment effects that do not rely on access to effect modifiers. We provided some general causal identifiability conditions for these parameters and derived causal machine learning estimators that allow us to estimate these these estimates quite flexibly in both randomized experiments and observational studies. We then also went a step further and showed that the properties of these estimators are actually quite desirable, at least if you're familiar with the causal inference literature, because they're double robust and asymptotically linear. And finally, we showed that it can be used or paired nicely with CATE-based approaches in terms of a sensitivity analysis to assess whether or not CATE is truly not finding any effect heterogeneity because there's no effect heterogeneity, or because we're just missing some key effect modifiers from our data. And so with that, I'll thank you for listening. And if you're interested in reading the paper, it's available on my website. I also have an open source R package if you want to take this for a spin. It's a little janky, it's a little rough. It has to be refined, but I'm very interested in getting feedback. So please let me know if you want to give it a try.
Okay, I'm going to ask a question. Thanks, Philip. That was a really excellent talk. You have a talent with sort of walking us into the weeds where I could follow, and so I appreciate that a lot. So two questions. One is, early on you listed, use cases, like obviously late stage de-risking was one, maybe that was the genesis. But does this also translate to risk factor epidemiology, or it's just too big, the assumptions of get too risky or unlikely?
That's a great question, and thank you for the very kind words. The truth is, I think it can be useful in plenty of settings, but it comes down to whether or not you know for a fact that heterogeneity is present or not, right? It's like if it's risk factor epidemiology and you know based off of your own clinical expertise that there are patient subgroups and they do experience different benefits from treatment, then maybe it's not worth going through this process, because you would automatically kind of negate this null hypothesis based off of clinical experience. But if you truly don't know, then it can still be used, right?
There's a lot of scenarios where I don't think we know.
Yeah.
And so, because we are exploring the TMLE in the risk factor epi space, but I hadn't thought of approaching this variance difference. My second question is, if you go to the slide where you had the two distributions, or well,
the visual intuition.
Yeah, here, it's very nice, very well done. Is there a scenario where, like, obviously this is in a counterfactual fantasy land, but I could imagine, like, you have these two identical distributions in the top right panel, but in all the examples of individuals, they were sort of, the delta was identical. But I could imagine a scenario where the distributions look like this, but at the individual level, there's a bunch of variability. So is that like an exception to the rule? Or
I did not mention that. So on purpose, I was hoping nobody would ask. No, I'm kidding. There's a statement over here that if the variances are different, right, then you cannot have homogeneity. This is true in all cases. The opposite is not true. Like, you can't go from here in this way. Just because your variances are equal does not necessarily mean you have a homogeneous effect. All right. You could think, because there are settings where you might have this kind of distribution and you don't actually have this distribution over here. But I'd say those are pathological in the sense that you almost always expect your potential outcomes to be related in some way. And if that's the case, then you would very rarely see that. The other case where this might happen is if your potential outcomes are completely actually unrelated to any kind of treatment. So again, it's like in pathological cases, you might see what you're describing.
This is better than the status quo of me not knowing anything about this.
Yeah.
Okay.
But the bottom line is that although that of like the interpretation, because if you see a differential variance equal to 0, then you want to assume that there's homogeneity. But the bottom line is, like, this is really what we're testing about, right? Like, if you do see different variances, then you're guaranteed to have a heterogeneous treatment effect. So I'd have to work out the conditions for, when what you're saying is true, and then they're probably reasonable conditions to, like, assume away.
Like a discussion limitations sentence that you threw in.
Exactly.
Jose.
So effect modifying is one way that you can have heterogeneous treatment effects, but there are other ways as well. It could just be that you're measuring the treatment effect with error, or it has nothing to do with the underlying covariates. Can you tell the difference between the two?
Yes. So in this talk, I've simply defined a homogeneous effect as potential outcome under treatment equals potential outcome under control plus some gamma. In the actual paper, we go one step further and actually split up the definition of our potential outcomes as some function of W plus some error measurement, some epsilon. And so you could actually have a differential variance where the outcomes themselves on average are identical, but the error terms are different. And you would still pick that up with this. But for simplicity, I just didn't.
Yeah, but you can tell the difference between. Okay.
Yeah, because essentially what happens is you would still have different, like the variances would still be different over here. You'd have like different spread. And so you'd still have this in practice, although it might just be due to like noise and not due to like an effect modifier driving that. It's arguably still interesting though, right? Because if you have, for example, two treatments that don't have like proper effect modification through some W, but your treatment, your Y1 has super wide tails, then it might be that some patients, you know, are like to the far left over here, and that could actually represent a really negative outcome. And so as a practicing like physician, you might say, oh, I'm not going to risk having a negative outcome in my patients and only restrict my treatment to the standard of care because that's like a safer bet. Even though maybe you do have this kind of like shift over here where on average the outcome under treatment is slightly better. That's a great question.
So thanks, Phil. That's inspiring. So let's say with Frederick, we have the following question. You do this, you identify what seems to be a treatment effect modification. Then practically how you proceed. Do you start examining some baseline characteristics that make sense as per clinical intuition, and how many of those you could explore, and what are the sample size limitations that you will run into?
That is a fantastic question. I've shown this in applications in sensitivity analysis. I don't have to worry about what comes next. But the next steps that I'm looking to explore right now actually are how do I use this as a go, no-go decision? Before I actually try and apply a CATE or I look at subgroups, can I just run this to see if there's any evidence of effect modification? And if there's not, fantastic, I'm saving myself a ton of time. But if there is, what do I do? What's the best approach? How do I make sure that I could step into the second analysis and not be penalized for false positives or false negatives? So like, there's still a lot of development there. I don't have a good question for you, but I am thinking, or good answer for you, sorry, but I am thinking.
Thank you.
Thanks, Philip. Sorry, I missed a lot of your talks. We had another meeting, but just expand on what you were talking about before with the lower left, the graph on the lower left there. Have you thought about essentially, you might call it quantifying risk aversion and bringing that in? In other words, how much negative value you would assess to a, let's say, a poor outcome with treatment, even if it only occurs in a small, relatively small proportion of people, and sort of building that into your outputs in a sense?
That's a great question. That's something that I'll have to work on with collaborators who are in the field working with like the specific treatments of interest in patients, because I think it's super indication specific. And also, like you're saying, like risk averseness specific, both on the patient side and the treating physician side. But yeah, definitely, right? Like again, like how do you, it comes down to how you interpret, I'll get to these parameters over here, right? Like if the standard deviation, if this value is large or this value is large, but the treatment effect is really tremendous, like your AT is tremendous, how do you factor in the difference in the variances? Do you always prefer the ATE as being like the guiding light, or do you consider the fact that there's a super wide, or there's like, you know, large tails over here? I don't have an answer, but something that's interesting.
As you say, it's totally situation and context specific. But I mean, from this sort of decision analysis literature, there is an approach to this kind of thing in general terms. Yeah, thanks.
Awesome, interesting. Thank you.
We do have one question in the chat from Michael. How would you explain how to interpret what a large difference in standard deviation? It's a lot less obvious than a difference in means.
Yeah, that's a great question. So I'll leave this slide up. So the difference in standard deviation. So it's with respect to this causal estimand over here. Really, what it means is like how different are the spread in between these two distributions. And so if you have a very large value, it means that and it's positive, it means that your treatment, the potential outcome under treatment has a very large spread relative to your potential outcome under control. Similarly, a very small value means that your potential outcome under treatment is quite small relative to your potential outcome under control. So that's how you think through it. You can really just visualize these distributions as how basically fat they are, how spread out they are.
Are there any other questions in the Zoom?
I think there's one more question in the room.
Hold on. Wait.
Thank you for the nice presentation. I might have missed this in all the statistical explanation, but in comparing the standard treatment against the neutral drugs, what are the safety outcomes considered in this case?
The safety outcomes that would be considered? It really depends on the indication in particular. So some indications have like gold standard safety outcomes that you want to report in every trial. Others don't. So I don't have any like concrete examples here to give you. But maybe some of the clinician scientists in the room could give examples of the safety or tolerability outcomes. Okay. Oh, so yeah, that comes down to what you know about the outcome itself, like the units. So like you kind of have a sense of what a large standard deviation is. And then you could also rely on statistical output from the estimation procedures by looking at the standard errors. That could give you some idea of like what constitutes big, at least relative to statistical significance, with all the caveats of p-values. All right. Well, thank you.